right.
We good?
We're good. We can start.
Okay, welcome everybody. So today we have a special guest coming from ASU, Banu Ozkan. Hopefully I'm saying that.
Very good.
Okay. She is a theoretician. She's working in the dynamics of proteins and interactions and so forth. Has some really interesting work. And yeah, without further ado, please welcome our guest today.
Hi everyone. First, I want to thank John for the invitation. I always wanted to visit NAU, so it's really fun to be here. And I know it is towards end of spring, yes, beginning of the spring break. It's end of spring break for me. So thank you for still hanging on. So when I look at the research in the department, it's slightly out scope of what I do, but hopefully you'll enjoy why I'm very much interested about this thing. So since it is really very different from what you guys do, maybe there is a lot of things I missed in my talk, which is very obvious to you, but not obvious to me, but not obvious to you. So I like it to be like very informal. I don't need to finish all my slides to give the whole story, which I'm very excited, but I don't need to. Interrupt me anywhere if things doesn't make sense or you get excited about a point. Just ask any question. Please do. It will be much more fun than just me doing a monologue by myself, right? So today, what I want to talk about is protein evolution. Also, this phenomenon called Allostery, which I will explain a little bit what it is, which is really important, through protein conformational dynamics, which is my expertise. So now close your eyes and imagine a gadget. That gadget doesn't need any resources, can make its own materials, can make its own energy. When it is broken, know how to fix itself. It can repair. And it's very sustainable and know how to clean his own waste, right? And when it knows that it is time for him to not doing well, he can remake itself, recopy the brand new one. What is that gadget? We all have, actually. Right, that's right. It's our cell. Exactly. Oops, that's the thing it always does. So that's our cell. Our cell is exactly like that. But every single process I told you that does is because of proteins. Proteins carry out all these processes I told you right now. And it is interesting what they do, for example, their workhorse of cells. For example, they make their own materials doing this enzymatic reaction. There's a reaction center surrounded by a bunch of other organic molecules, and it makes a reaction that takes million years to make millions of them in a second instead. Extremely efficient. And then what they do is they also make their own resources, as I said. They do this energy unit called ATP, and making this molecular machinery. That molecular machinery is very interesting. It looked like a little rotor model, right? And some reaction, proton pump, makes it rotate actually. And if you look at the efficiency of this rotor model, it's 90%. So nature made the most amazing, efficient nanomachine in the world that we can never manage to make the exact copy of that. Why is that, right? Is it because they are jiggling and wiggling? That I'll come to soon. And finally, like sometimes, if you want to carry something inside the big cell, like say in an E. coli, right? It's limited by the diffusion rate. So you have to bring these molecules to where it's supposed to be so much faster, even in your brain, around the shops. You have to beat that diffusion limit. How do? They create this machinery called motor proteins, and they also make from the same proteins like train tracks called microtubules, and these proteins move very efficiently, in a sense, and know which direction they go. They are also built to know which direction they go. They can only go in east or west, but not both, and does that. So they're built very effectively, very amazingly. But if you look at that, wow, how they did, what is the secret in this thing, right? When you looked at that, you see that proteins are made of amino acids. This is biology 101. Maybe you took, maybe you don't. But there is a specific, there's 20 types of amino acids, but there's a specific sequence of that amino acid come together like a sentence, right? When you read this is a sentence, it makes so much sense in cells when this is encoded, and it makes maybe a motor protein. Another sequence make that rotor machinery that you saw, right? And if you look at the chemical composition, they have the same chemical compositions. Like there's a central alpha carbon, then that has 4 bonds, which are hydrogens. And there's this dangling here, which is, we call it side chain, that gives a chemical specificity. Like if you have those oxygens, right, it is more acidic, so it's a charged group, like, right? And then sometimes you are polar or non-polar, or it can be like, instead of, it can be finally positively charged too. So like those different sequences, when they come together, they make this backbone, which is same, and you have those more like non-polar group versus a polar charge negative group and so forth. You end up this huge polypeptide. Obviously this polypeptide not gonna stay like this, right? All extended. For the PhD students, why is that? All stretch is entropically not variable, right? Right? Like every elastic materials, it has to compact. It gets compaction, but when it's get compacted, it doesn't get randomly compacted. Instead, the build of interaction creates like a little, know which interaction should go on, that it creates like a network of critical interaction, like a unique 3D structure. So we call that thing called protein folding. So what happens is, in split second, that protein goes, and so this was a big deal before. Now with the fistus things, we can't fold any proteins now, but I'll tell you a little bit more that. Like comes very organized pack unit. This sequence go this way, but another sequence doesn't go that way, all right? So, and one thing when I was graduate student was driving me was how they fold very fast, and why that sequence get that specific structure was the question I want to answer. And turn out that we call those functional state native states, all right? That's, it is only in that structure that protein is active and functioning. If it doesn't fall to that structure, it starts lumpy, sticky hydrophobic chains, and all those hydrophobic sticky chains compact together, and you get an aggregation, and it leads to a disease. So your body, your good cells, if they're not aged and senescent, know to fold quickly. And if not, there are other organisms help them to fold quickly, but only sequence make that. It's because in our earlier work, in my PhD, we show that the folding landscape looks like a funnel, where the native state is very deep down and energetically the most global minimum free energy state, right? But if you think about it, the whiteness is related to entropy, conformational entropy. The depth is related to enthalpic interactions, all right? So the more you gain interaction, so it is like a folding is a war between entropy versus enthalpy. So when you gain strong interactions, all right, you gave up your entropy, but those interactions are so strong that's built in this that beats that entropy and make this. That's why when you don't have many interactions, you have so many degrees of trade, which is like a funnel. But the good thing about being a funnel is it makes folding fast. right? Because wherever you start, there's a downhill that will go, right? Like think about if there's a huge fall with the funnel, if you throw the ball, stochastically, it will end up there, right? So that was also, we explained how this happened. But the story was not just that, right? So, okay, this is how it falls, and there's a 3D unique structure, how that structure look like, right? Like in 50s, after the discovery of DNA, all the scientists says we want to get that structure of proteins, and it will be beautiful, and we'll learn so much about it. And they get the 3D structure of DNA after DNA protein. That was Max Perutz, who got Nobel Prize because of that. But it was a shock because yes.
You said that this folding energy is all downhill, right, towards the global minimum. But like 2 slides ago, you had that simulation where it looked like it went into a local minimum.
Right. So yes, very good point. Let's go back. Good question. So here I was always showing a slice of that landscape, right? So you have, although it is funnel-like, you have like this, sometimes this not global, like there's also some minimums, like those kind of intermediate states that you can get stuck, and there's the barrier to go down to that actual global free minimum. But because those are fluctuations and they are in a bed, a random kick happened to jump up and go down that. And indeed, that was also in 90s and late 2000s, how those intermediates and the barrier separating, is it like the folding landscape is like a glassy dynamics, or it is more smooth, that the barriers are not, how rugged, right, that funnel look like was the question. Turned out that the rugged is like optimally smoothed out, meaning that majority of interactions that take you to native states, we call them native interaction, are usually so much stronger than a non-native interaction. Let's say in the sequence 5 and 10 needs to come together. That attraction is so much stronger than 5 and 25, right? If you make 5 and 25, you need to break that first and then make 5 and 10 to go to folding state, right? So turn out that folding landscape is minimally frustrated in the glassy dynamics work, right? Right, so it's very compact, but it's also very ugly, right? It doesn't look like that beautiful DNA. But then they go through, then the field of structural biology initiated, and they said, okay, let's try to do some data mining and understand what's going on. The first thing what they did, majority coming from polymer physicists, they say, okay, let's look at the dihedral angles. I don't know if you know this, but when they look at the structure and dihedral angles, turn out that there are certain preference of dihedral angles, which makes like some helices, and some of them are more planar strands, so they like organize and give that, like there's some local contacts formation, like amino acids that are close in the sequence come together either like a helical turn or more planar turn and make hydrogen bonds. That's what we learned. But one other thing turn out is it's not the structure which is important. Turn out that what was important is once it folds, it's not like a static structure. In that native val, it's still tiny bumps. It's exploring still certain degrees of freedom, and actually it is jiggling and wiggling, and that's what Feynman said. If you want to understand what jiggling and wiggling are, then you will understand how they function, right? And why it is all jiggling and wiggling? Why it is because everything is fluctuating, because it is in a thermal bath inside the cell. It has all crowd materials and then water, or in your test tube you can do that too. So, and if you look at that, there's even everything is jiggling and waggling. What you can also notice that there's also like more collective motion, like all those secondary motifs, local interaction come together and do like a collective motion, right? Like, and turn out that that's very important for the function, right? So this is all observation. You can do this type of observation, but how can we understand and say, read that sequence and say, I know what it is and I know what this function and I can change this and design new materials using proteins, right? That's what I want to do. And at that time, let's recap what we explained so far, right? So if I'm teaching a class, I'll ask you guys, but instead I'm going to tell, right? So proteins, this is called made of the specific amino acid that dictates, right? Here, every amino acid is shown by a single letter, right? It gives a unique three-dimensional structure based on that sequence, which is unique, different than another structure. But that structure has a function, but the function happens because of dynamics. And if you see here, that dynamics is opening and, let me do this again, opening and closing. And that opening and closing, for example, pass through ions here, for example, in a membrane protein. So, and it built really things, and there's some charge and electricity interaction opening and closing that dictate that ion to pass through your membrane, for example, right? One thing interesting is if you make many changes in different random sequences, like you say, I can make this and play with it, just take this amino acid and pick from your bag of 20 different amino acids, put another one, put randomly. It's very robust, it still functions. That's what we need, because when our body cell copy itself, it can make errors and your protein is not exactly the same. Then if your cells don't function, you'll die, right? So there is robustness built in. But then sometimes environment change, you need to adapt to new changes. So that you need certain specific changes that makes this, for example, function in a higher temperature, because you're suddenly Earth got super warm, right? We don't know who will be that lucky cells who has, but anyway. So that's what proteins does. And how, where, it's very difficult. The only thing that's been solved is nature solved that, right? Through 3 billion years of evolution, all right? So if you look at from today to 3 billion years, you have many species, right? And all these many species has the same proteins, right? Like we have that protein carry oxygen to our cells, which is very important. But it's not just us. Bird has it, right? Chicken has it, hen has it, fish has it. But there's slightly changes too because of different species, right? Because of their condition, environmental condition, they adapt, slightly changes. And then, and this happened through, I said, 4 billion years of Monte Carlo steps, right? It wasn't really a great optimization. Some copy error carry over and you become lucky, those things and stuff. So what I would like to do is try to use evolution sequences, because nowadays you can have every species genome and those proteins. It's so nice and do comparative modeling. So what's that I do? But I said, I'm not an experimentalist. So how can I understand those interactions that are critical, right? Thankfully, I didn't need to do this. So some former people, much more older than me, developed force fields to study that underlying landscape and energy interaction of proteins. Indeed, that was the Nobel Prize in 2013, right? That's been given to the 3 gentlemen here from chemistry, but they were all physicists, by the way, all right? And so there's the molecular mechanical force field, and also then how you understand that jiggling under that molecular mechanical force field. They built a software or a principle called molecular dynamics simulations. So I want to briefly explain what it is. All right, that's very important. And the 3 guys get the Nobel Prize. I personally know Michael Levitt pretty well. I used his slides to really, I think he also used the same slides in his Nobel talk to explain what it is, all right? So So what you need to do is you come up with bounded and non-bounded interactions of a macromolecule, a polypeptide, right? So if you think about it, so we are looking for some internal motion. It's not like as a system you move in 3 dimensions. This is how they're jiggling, like the internal coordinates, internal motion that we are interested, right? For that, you can have maybe a little bit like a bond stretching, right? If you're covalently linked, and that bond stretching can easily be represented by a simple harmonic potential. Likewise, if you have 3 atoms that are covalently linked to each other with 2 bonds, right? That can be a also motion like this, which can be modeled from angle bending, right? So then you can do like angle bending, like also a simple harmonic where that angle is the proper angle given the bonds, right? And finally, this is the dihedral angle, which I was telling you. If you have 4 atoms that are connected by 3 bonds, right? A dihedral angle is the angle between 2 planars. I don't know if you can see that. Like there's one planar between these 2 vectors, right? This and this, right? Make a vector that you can have a norm. And another from this and this that had a norm. And the angle between that 2 norms give you your dihedral angle. So I usually bring, like, think about my hands like this. Like this is the bond, and these are this other 2 bonds. When I rotate that bond, right? The distance between these 2 far ends can change, right? And since based on, like, you can have this distance that way when I return to you, the distance between this 2 fingers, right, 4 fingers, or if I rotate that can be the same. That's why you can have this type of cosine function that you can represent, right? And actually in polymer physics, that is the softest potential that by rotating dihedrals, you can really recover a lot of conformational space and different positions of all those polypeptides. And finally, for the non-bounded cases, as I said, there are charges, right? Some atoms carry charges, so there should be classical electrostatic interactions that you show with this for the charges. And finally, you can like due to polarity, there are certain charges on polarity. You can, atoms needs to be like a non-bounded atoms want to be specific location, right? Short-lived specific location, right? You can use by van der Waals type of interactions, which you can use this Leonard-Jones. So those are the interactions. Obviously, you have to need the parameters, right? So we use quantum to get those force field parameters. So take every single atom like a C to C bond, identify charges, look at the stretching from quantum level, and you did all quantum level calculation to get those force field parameters. Only this part and this part a little bit needs tweaking by using the rotation degrees of freedom and some fitting experimental data besides quantum. And then you get the whole force field parameters. Now, then what you do is you want to use classical mechanics, very simple, to sample the conformation on that energy landscape. That's very simple. If you think about all we are using is Newton's principle, right? Which is F equals ma. So think about it. Let's do this landscape in 1D, right? You have those energetics, so only in X direction, let's say. So there's some, right? So that gives you like a landscape, which if you know your potential, right? Negative gradient of your potential gives the forces acting on this landscape, right? It is, so if you have like that energy landscape, negative gradient is the slope, right? Tells you, for example, if you're here, you most likely go down, right? You understand the forces acting on that. And you can do it 2D. In our case, we're doing in 3D, X, Y, and Z direction, right? Now, so since we have the force field, we can also calculate the potential U, right? We're taking X, Y, Z direction. We also find the force acting on a system. Like let's say we have those molecules moving around, right? So we know the force by the negative derivative, right? Law of the potential. Then we know from Newton that the mass times acceleration equals to force, right? Now, there is a problem here because everything is moving and jiggling, right? Everything is changing. So because of Heisenberg uncertainty principle, we cannot have an exact location and momentum, right? We can't do that. So there is no analytical solution that we can use. But if we assume that we want to understand, say, this guy, right, which will be his next acceleration and location and so forth, in a very tiny time step, say like 1 femtosecond, which is 10 to the minus 17 seconds, right? Then you can assume that those guys are almost the same. The forces acting on this guy after that is exactly the same. Then we can do a numerical solution, right? That's what we do, right? Then, for example, if you want to calculate the new position, you use the old position, right? And the old velocity, and calculate the accelerator from the force. Now you calculate the new position. Likewise, you can assign the new temperature, new velocity from the old velocity and the acceleration, right? With the numerical solution. And finally, since you know all the velocities, you know the total kinetic energy, right? That gives you your temperature. So that also gives you a sense of temperature, right? And the whole thing, you know that the total energy, which is potential plus kinetic energy, will be constant with time, right? To obey those things. And as I said, the time step must be very, very small, right? Yeah, actually go like very tiny step and explore that scale. It was amazing. This was built in 1980, right? And like 30 decades later or 40 decades later, actually, it's almost half a century now. It's amazing. Turned out that force fields are really great. You can do this for not only organic molecules, but you can do also any material, inorganic materials. You can do these things, and it's pretty good. You can run, not with the speed of computers now, you can run milliseconds. Imagine gazillions of steps you can go and generate. When I was a graduate student, they thought the first protein from the force field. It took 3 years. It was only like, I don't know, like 100 nanoseconds. Nowadays, in my lab, we run 100 nanoseconds in a couple hours with the GPU machine. So it's a different era now. It's very fast, and that becomes like truth. A lot of experimentalists, when they do find something, they also want to simulate to really understand what's going on, because it gives you a microscopic picture indeed. So they say computational microscopy, like something they also named this, and it's very powerful and useful. And I use this in my group to really understand those interaction and jiggling and wiggling, right? All good? All right, perfect. Now, sometimes though, if it is super huge, I call it super complexes, right? Then we hit the bottleneck. And then, instead of using all atoms, you can use a few atoms. We call this coarse graining. And turn out that actually something magically can happen if you do something very simple. And a lot of you guys probably got those harmonic oscillated and did normal mode analysis, right? Turn out that you can apply normal mode analysis to proteins too. So what you could do is, for example, if you know the structure and the coordinates. So what you're doing is you're taking, I always use this one, but it's not connected to this one. All right, let's do this one. So you take the structure, right? What you're saying is do you remember that energy landscape, that native well is so deep that you can fit a little harmonic parabola around that well, saying that any position, like on coarse grain, those atoms with central amino acid, right? And then at the little harmonic spring, those close in space in the structure, right? That's all you're doing, right? You can do this with NO, right? And write a simple harmonic potential for them, for those interacting, all right? Once you do that, right, since this is a norm, and you're just making this parabola near this native value, which is global minimum, right? Your first derivative is 0. All existing is your second derivatives, right? Right? So you can write a matrix of your second derivative from that function. And simply then your mean square fluctuations and covariance of each fluctuation is nothing but inverse of that Hessian, that second order derivatives. And this is analytical. It takes only 10 seconds in your computer. And it turns out that if you're only interested in square fluctuation of one protein and a different protein, completely different form, it's very powerful and it's very useful. And we use that and we get those collective motion, which is important for function, very carefully done. Right. So therefore, in my group, we use these two different methods very well. And actually, as I said, this was developed, pioneered with my mentors when I was a graduate student. So therefore, it was like, baby, for me, I have to use all the time. All right. One thing I also add this is, like if you do experiments, when you move something, you also give a little perturbations, right? And usually perturbations poke like a random force, right? That gives like how things, like when you poke something within the jiggling, how it propagate happens, right? So I want to simulate that in my analysis too. For that, what I do is I put I say when I calculate those jiggling, I say given this covariance and jiggling, if I give in little Brownian noise, how that jiggling propagate, I want to look for that. And for that, I use linear response model. So it is interesting, for example, here's a protein. It has two states, and one state is inactive, other state is active. Once the protein folded, it can go between these two states, and once it's active. And if you give a little, for example, little jiggle to one end, and calculate the response of everything, like I give that, you see that it is making already, the first collective motion is already pushing that to that direction, like the landscape is already built, go from one length to other things I can compute. So what I do then is, this is what I do, either I run a molecular dynamics simulation or run this elastic network model and calculate the covariance. Once I calculate the covariance using linear response model, which is the equation, but you don't need to worry about that much. If like give a minor random Brownian kick, right, it will propagate, and because of things, induce certain normal modes and find its way, and everybody feel differently in different direction and different magnitude. So I can compute this response, and I scan like first one, second one, up to every position one at a time, and calculate a response matrix. Then I calculate this thing from that response perturbation response matrix, which is like the total fluctuation response of a position I when the rest of them are perturbed, right? And I name this matrix dynamic flexibility index. This is interesting. So here is, I did for this protein, and I plot its ranking here per position. This is first positions, 80 positions in the sequence, how flexible, how responding to that perturbation happen any part of the chain, right? And I color code that in the shape of the structure over here. So if you look at this, this is the core of a protein. It is all buried positions are here. This is exposed to solvent. This is exposed to solvent, right? This is and if you see that there are certain positions super rigid, I put 0.2, and certain positions very, very flexible, like this helix is moving so much when you perturb, for example, that red helix over here, right? So if a site is super rigid, very low, it means that within the network, when it's moving, it has a lot of interactions, right? When it feels it quickly, it does, because a lot of interaction, it doesn't move much, right? But transfers the rest of it very quickly, like your elbows, right? Or a hinge of a door, right? And based on this analysis, I assume evolutionary, because they're very critical for the motion, collective motion they're controlling, right? They should be important. And importance in evolution means that if you mutate, that's a dead end. It should be conserved, right? If it is mutate and it is that, evolution eliminate that, right? So this should be the sites that are very conserved during the evolution. If it is very flexible, high DFI, then it has a lot of degrees of freedom, right? It has a high entropy. So the changes, if I have high entropy, any changes you do, I can absorb because I have a lot of degrees of freedom. If you put a constraint changing that, I find my way, right? That evolutionary, then it is okay. You can make changes. And evolution thing, it's okay. You can keep that, right? And that's what I checked first. And so, because my goal was really understand the principle in evolution, right? People use this actually. Francis Arnold, who got Nobel Prize, mimicking evolution in a lab called directed evolution. Like if you want to make a new material, a new enzyme, it make random mutations and just select those doing function that he likes a little bit, she likes, and keep doing random shuffling like a little black box. My goal is really understand what is filtered in that black box, right? First thing is it really the dynamics, very big criteria? And when I did that, I learned, number one, evolution really uses protein flexibility for the emergence of new function. Let me explain that. So for that, what I did is I take all the protein structure of human proteins. You wonder why human proteins? Humans are the last evolved species on Earth, right? I'm not saying the most, I'm saying last. Because some people make fun of me when I said most. Anyway, last evolved species on Earth, right? So then, the same protein is, a lot of other species has the same protein, and you can calculate rate of evolution using those species because you know their emergence in time per position. So I got this 100 protein, roughly 40,000 positions. I calculate their flexibility, bin them, and look at their evolution rate. And this is that histogram. This is percentile ranking, like very rigid to very flexible. Yes?
Reading that last, I just want to get that. So you're saying that there's nothing that has evolved from us?
So far, yes, we don't know, right?
But isn't most, or a lot of species that way?
No, but there is a, okay, good. Yes, all the proteins are species right now on Earth is in their genome things. But if you look at the emergence of the species, this comes from Newton's theory of evolution actually. You can predict the emergence time. Obviously, E. coli come on Earth so much before human, or fish come on Earth so much before human. That's why I say the last emerged, let's say, instead of evolved, okay?
You had that tree diagram, that, is it called phylogenetic?
Right, I'll
tree?
show now, yes.
I mean, maybe pointing him back to that
Right,
would
yes.
show where mammals and finally.
Yes, that's a
primates.
good idea, right, let's go back.
Sorry.
No, it's a great idea, let's look at that.
We're last evolved relative to all other mammals too.
Like,
So here is the thing, right? Look at the bacteria, right? Bacteria emerge compared to human, right? That's the phylogenetic tree, shows you their emergence time, right? So humans emerge so much later, or most recently, right?
you see Neanderthals
Right,
at the moment.
right. We're compared to E. coli, for example. Okay? Okay, hold on. It's giving me a hard time to go that way, sorry. All right. So here's the plot. Let's go here. And So what you see is, right, if you look at their very rigid ones, right, that also their rate of evolution is very low. Very flexible ones, they're highly evolving. Indeed, I didn't show if you look at poor correlation, that correlation is 0.86. That really proved that that is a very strong filter, the physical principles that the nature uses to do it. Now, what I also do is something quite interesting, is I use this analysis to study ancestral proteins to see how function emerge. So what I mean is, let's look at a simplified version of that phylogenetic tree. If you look at, this is E. coli, right? A human emerged so much later, right? And you can look at their ancestral, and this is the most important thing in evolution. And actually that's, some people say that's the most important information that we should carry over if all the knowledge around Earth is disappeared. You heard about that, right? It's the subject where Feynman asked that, and you know what Feynman thought, right? Feynman said the most crucial information is everything is made of atoms, right? But evolution biologists thinks actually it's also very crucial information is everything comes from common ancestor, last universal common ancestor. We call it LUCA, right? Right? So it is very hard, so this is the phylogenetic tree. What people usually do is, you have this human, horse, fish sequences, they go look at that. But these are, as you pointed out, these are recent species. Comparison in that whole branch is not that understand the molecular adaptation of the process. Instead, we need to go down to the root, and that's what I do. Obviously, it is very hard to get the ancestral sequence from fossils. Instead, you can use nowadays mathematical tools, because since we know every sequence and we know how they linked from that phylogenetic tree, you can create a Bayesian frame, a mathematical model, saying that, for example, if human and horse are coming from same ancestor and these are the sequences, what is the most likely sequence of their ancestor? So you build up like a Bayesian frame to look for hypothetical sequences that represent those ancestral sequences. And that's what we do in our lab. And then you can also make those proteins then in your lab and test them. Interestingly, as I said, if you randomly do something or make mutation, they're all functionless. You're making these ancestral sequences that there are hundreds of differences from the extant one, they function. So, and not only they function, they do something very different. I did many of them, I'm just gonna give you one example here. This is a protein called beta-lactamase. Beta-lactamase is a big friend of bacteria, enemy of humans. Why? Because when you have a bacteria in your body, it's really bad. What we do, we pop up antibiotics, right? If you have a swollen throat, right? It's really bad, high fever, you get your antibiotics, everything goes well. But because you're killing the bacteria, so bacteria has to have a defense mechanism. And that's what they do. They make this protein, shoot the membrane, and then this protein is an enzyme, chop the antibiotic. It's called chemically dehydrolase the OH bond of a hydrogen bond of the antibiotic, all right? But so what we did is we take different bacteria and predict the sequence of gram-positive bacteria, and this is the gram-positive and gram-negative bacteria sequences, and this is the ancestor of Enteria bacteria, all right? So one thing interesting is when you make those sequences and characterize in laboratory conditions, this is the current ones, and these are the ancestral, like they happened like roughly 4 million years ago. This is the denaturation temperature. What it means is if you make a protein in a test tube, it functions like in cell, does what it's supposed to do, but it is in ambient conditions. when you increase the temperature, right, how you cook an egg, it denaturalizes. It's no longer in that native fold anymore. We call that denaturation temperature. While the current bacteria, TAM1, has the denaturing temperature of 40 degrees centigrade, right? That's why when you boil something to kill, you boil it, right? And it dies. For ancestrals are like dying at 90 degrees centigrade. They're amazingly thermally stable. The hypothesis is maybe 3-4 billion years ago, Earth was very hot, super hot, so you have to survive in that ambient conditions. That's one hypothesis, but it is hypothesis still. And the other interesting thing is, to me, this was like shocking to me. You probably heard about antibiotic resistance, like now antibiotics is evolving, and now it is the era that suddenly with antibiotics will work, right? Because some bacteria know to kill many different antibiotics. They gain many functions, right? So if you look at this, this is the turnover rate, how fast, how quickly these proteins can degrade different antibiotic. Plot is here. Let's look at the old one. The old one here degrades penicillin, right? That's the first discovered antibiotic. And with the same efficiency, it degrades another antibiotic, blue one, which is called cefotaxime. Cefotaxime was introduced 70 years later, the discovery of penicillin. Amazingly, old guy knows how to deal both of them already, right? To me, it was promiscuous already. So then that was built in it, actually. So that, but what happens is probably, that's also, when we start bombarding this guy with penicillin, because it gave up its degradation for penicillin, but learned to degrade super faster penicillin instead, right? So there was a emergence of like doing something floppy of many jobs, that excelling in one job only occurred through the substitutions, right? When you look at their crystal structure, it's identical. Nothing has changed. Oh, I said there's a real reaction center, that quantum things happening there, really. There's an electron transfer, bond abscission. Let's look at that center and type of amino acid there. Identical, nothing changed. How did they achieve that is the question. Turn out that if you look at their flexibility profile, you see that there are certain parts becomes very rigid, and certain parts becomes very flexible with the substitutions. Indeed, if you map them in the structure, it makes more sense to me, because this is where the antibiotic binds. An old guy has much more flexibility, so accommodate different antibiotics and induce that changes to get that catch, right? And when you gain function, you want to excel, you become like a lock and key mechanism. You just recognize only penicillin very fast and this is how you excel, right? But there's always the thing is, you want to do this by, that's what we learn, is you invented the wheel, which is that fault responsible for that function. You want to keep the fault, so when something is becoming flexible, flexibility is also always introduced, right? To keep the fault, there's other part, compensate and rigidify. We call this like a hinge shift mechanism, right? So like some hinges, you lost a hinge, but at the new hinge, now you have the new normal modes for the new function in a sense, right? So we see those hinge shift mechanisms with all the evolutionary systems. Actually, we think that's a model, like shifting normal modes is a way to modulate function in protein evolution. And this is nothing but called, discovered earlier and named second secret of life, allostery, right? Now, I don't have too much time, but I will end up this story maybe, which is, I'll give a really crash course on about allostery. What is allostery? Allostery is, let's say you have two states, R and T, relaxant state, right? So let's say this was discovered with hemoglobin binding oxygen, And if you ever take statistical thermodynamics class, right, you have to understand average occupancy. This is a model I always ask, like if something binds, if there are two-state systems that they're binding, if it's a stronger, you add the additional coupling, right, that increase the average occupancy so much stronger, right? That's, I don't know if you did this, but you can do this without adding coupling using Allostery. What I mean is, let's do this as a model. Let's say you have two states, R and T, that you can sample, where R is more stable than the T, right? But when you bind, your stability change, but the T state is more favorable for the binding, right? And if you do the math and write the all Boltzmann weights of those states and write the partition function and calculate the average occupancy from the derivative of partition function with respect to chemical potential mu, you see that indeed you create this cooperativity term. Allostery is cooperativity. With the tiny bit of ligand, you suddenly go all or none and everything becomes binding, right? And without adding any cooperativity term, this model gives you this type of idea, and we call this allostery. And turn out that you don't need to have a distinct two states. Later, this guy, Cooper and Dryden, showed that even shifted normal modes can give you this type of change in landscape, only shifting the normal modes, right? That was the idea. So, and this is a very simple model that shows that, let's say you have a scissors like this, right? Now, let's say you stuck that part. Now your normal mode will change and something come and bind on top, right? Then this part will be so much easier to bind and more, right? So therefore, while binding, we can also change the mutation and change the stiffness, right? In our normal mode and shift the mutations. So therefore, shift in normal mode's peak distribution, like you can do even binding, which nature does, or with mutations. That's the principle we figure out what the evolution does. Indeed, we computationally calculate vibrational density of states of these ancestral proteins, and this is the zoom. What you see is that the most collective motions, which are low frequencies, you see a shift their energy distribution in that range, really also prove that, and this is also show that indeed that principle is very important. All right, so with that, I will end up, what we also saw that using the same data, we can also, let me switch this. We can also, one thing interesting in protein evolution is this shift, certain part of the shift happens not because you make mutation exactly that side, you make a mutation very far side, it makes the dynamics change for another side. So it's like a remote control, right? Very remote control, I call it minimum perturbation. You don't feel what's going on, but you change slowly, maximum gain happens is a principle. For that, we use again this perturbation matrix, and we say, for example, what is the response of any position I when you perturb a functional site versus its average response? And if you color code this, for example, you see sites very close, always when you perturb this site, always these are very close in space, they will feel it. But sites super far away can also feel as much. And that way we can also calculate those allosteric shifting hubs and make mutations and design new proteins. So we prove that's true. These are the mutations that change function that we capture from this plot. And indeed, you see that highly coupled sites, functional sites, are the ones that are mutated. And I'm going to skip this part, but I'll say now, completely using this principle, we're designing new enzymes. And I'm going to end there instead, saying that it was very hard to predict those things. Looking structural evolution, there's a big gap, and we're hoping to gap it this way. With that, I'm going to skip all process that we do. not only we do, but we also experimentally show that with this design we can change dynamics from NMR. Also shows that we are shifting dynamics to what we wanted. So I will stick like that. This guy is called, his name is Banu, who discovered the allostery. At the same time, he also modulates elucidation of gene expression, how different genes turn on and make different things. And he got the Nobel Prize because of his first work, the gene expression, but he always said actually allostery, which is much more crucial for life. He named it second secret of life, and when I work in evolution, I see he was right, so therefore he's my hero. All right, and with that, I just wanna skip these things and thank to my group who did a lot of work. I have a lot of collaborators really helped me to have that experimentals all around US, even in Spain, and also the fundings we got. Without funding, I wouldn't do. And thank you so much for your attention.
All right, thanks. Do you have questions?
Yeah, I have a question. I just want to make sure I understood something. So you were showing this DFI. That was just for the human sample of humans?
No, you can do for the first time I did for human proteins, but you can do for anyone, any system.
That was just showing the variation in.
I did that for human proteins to show the proof of concept, because you can accurately calculate evolution rates, because human proteins has a lot of mutations also. Like it's been the most sequenced species on Earth. It was very useful as a good data model to work on it. Yeah.
So I'd be curious. Have you looked into, comparing? Because you said that fish and birds have a variant of hemoglobin. If you were to compare humans with more distant ancestors, did you get better overlap, or with that? I'm just curious.
Right. You see a gradual shift in their DFI profile. Indeed, when you cluster the DFI similarity of those flexibility profile, you see they are exactly clustering the way their biophysical properties. That's why we do in design also. So we make changes and then look at those the moving plan. There's a flexibility profile we target, DFI profile, and we want to push this profile to that profile with the mutations. Yeah. Yeah.
So I know like a lot of proteins often are like dimers or like octamers, like histone. When you're modeling these proteins in the way that they move, you have to look at that whole like octamer or dimer.
Right.
Or can you just look at one subunit and get another?
That's a great question. It is important to look at its functional form. If the functional form is a complex, as you put it, like two together or dimer octamer, then you need to do dimer, because the whole motion will change, the flexibility profile per sequence will change, so you have to do that way.
Yeah, so I was wondering, you have that evolutionary diagram where, went back to the horse and the bird and stuff. There were those two branches that converged off to the right, like long before. I was just wondering what those were.
Let's do. Sorry, let's do this. All right. Here we go. So.
Yeah, so off to the right, like before that big circle, there are those two branches.
Oh, I see. Yeah. that's a different kingdom, I think. Right.
Yeah, I was wondering like what's further away from us than like a coli. That was kind of weird.
Right. Yeah, like because plants and stuff, there's all three kingdoms, they come together. That's a different kingdom that we think.
They're not the archaea?
Yeah.
Archaea bacteria?
Yeah. Yeah.
Those are the really high, you mentioned them, I think, as high temperature thermophiles.
Right. So interesting, yes. some of them are thermophiles. But if you look at the thermophiles of today and the ancestral proteins 3 billion years ago, thermophiles are not as stable. So it is an interest, and they are not emerged, like thermophile is not emerged from those things. It is a different evolutionary thing. Like, so Earth cooled down, things change, then these things move to some other systems and they adapt again. So that's how it like happened. Yeah.
The question is about these simulations. Are you using on classical approach or are you using like quantum-based approaches as well, like at the fog or the
Right. In my simulation, it was completely classical. But if you really want to understand how motion impacting the reaction, the coupling, then you can do like a quantum MM kind of a thing. There is a mix, but I don't use in my thing because I really want to like I said, I want to take that sequence, which I don't, and make a numeric value from those dynamics and say that's how I did it. That's what I'm doing in a very simplistic way, right? That's why I don't use the quantum thing.
So you are not calculating the entropy?
In a sense, I am calculating entropy per site in that packing thing. That's exactly, it's related to entropy, like configuration entropy per site is being calculated that way. It's related.
And would you say that the entropy is the dominant factor in this?
Yes, I think whenever I work on life, it's the entropy, that everything goes entropy. It's the maximum entropy, the direction things replicate, like, it's, yeah, every time you hit that, and yeah, it's very important. And you had a question?
I wanted to know the types of human protein that you use. Maybe you use several times and then you came out to a conclusion, or you just had a specific one.
Can you repeat again?
I just wanted to know, you're using human protein, so I just wanted to know the specific type of human protein, or you use several times and you came out to the conclusion.
Yes, I use many different human proteins. I use the human proteins that are essential to whole kingdom, like everybody has that protein to really understand the evolution rate, because I want that protein shared in different species, right? Like there are they call them in biology essential genes, right? If you don't have that protein, the cell can divide and turn into a different organism. So that helps me to really calculate evolution rate properly. You had the question on. Oh no, yeah.
Are there any real water molecules in your simulation?
Yes, it is, yeah,
Okay, I'm sorry I missed.
yeah, we have the real water molecules, and solvation is so important, you have to model it accurately, and a lot of things went through how to model proper solvation, actually, that was a big field, yeah. Obviously, I'm not developing, but I'm using the best ones.
I will ask a question. No, excuse me. So usually when I think of this evolutionary movement of proteins through time, you think of these homology models that compare sequences, and it sounds like you're using an element of that, but your DFI score is kind of a unique perspective on the idea of protein evolution. I would imagine between those two methods, different residues are going to emerge as important or less important, so conserved or not conserved. I'm wondering if you found information.
So homology models, what homology models use is because if your sequence is very similar, right? So there's a map sequence structure. If your two sequences are more or less very similar, that you get most likely the same structure, right? The whole idea turn out the machine and all artificial intelligences how to pick those similar sequences, because it was very hard. Sometimes homology doesn't think they're homolog, but turn out they are somewhat homolog because they have the same structure. So that that idea, I see what you're coming from, is that you make that prediction, so then you can make an uncertainty, right, per site, saying this site is I'm not sure about it's the right, the homology doesn't give me a signal and stuff. And you're saying, is that signal correlate with somewhat flexibility? Yeah, on certain degree it does, but not too much. And also, like when you simulate, for example, you also look at like fluctuation jiggling from the mean. You can easily calculate, but I do all effort of this perturbation, all those things. The thing about is that I do that way of perturbation because there's a lot of coupling going on with different, even long range positions feel from different, like when you perturb this, some mode comes and you're very coupled, right? There's this long range coupling pairwise or sometimes triple things. So those are also important for flexibility stuff, which is those signal threshold ratio in homology becomes weaker and they end up not that accurate. But there is in the first order principle there is some correlation. Right.
Any other questions?
A final comment, probably incomplete. You know, we learn in the textbooks in physics that entropy is equal to disorder. Okay?
And yes, disorder.
No, however, when you have, for instance, 4 metals, you are doing an alloy, you are increasing the number of metals, you are increasing the configuration of entropy. But that doesn't lead to disorder, that gives an order. I mean, you get order alloys sometimes.
Right.
So the way I see very similar, entropy is not really disorder, it's really diversity.
Correct.
So when you have
Yes,
diversity,
that's
and
very
then
important,
that's,
yeah.
it might be ordered, might be not ordered, but it's really.
Right.
Okay.
So maximum entropy is a principle, how we enhance diversity, and that's the key thing in life, the nature use that. You're completely right, and just to add enough, and not only you add this diversity and how you poke something to enhance diversity, this still thinks is the Allostery, that suddenly you do a lot of changes and make function. Nature figured it out, but I said that was a constraint. You want to keep that structure as it was very hard. You know what nature said? Forget about the structure. I never told you that. There are proteins completely intrinsically disordered, meaning that they have no structure, and more evolved species like humans compared to E. coli have those kind of things. Now you get this diversity very quickly, and if you modulate something, give a signal, then signaling proteins are usually disordered. So you were right about that, and that's exactly which we are also moving forward in that regard.
That's just one little question. It's clear you made an enormous amount of progress, and the whole synthetic biology front that you mentioned is evolving very rapidly too. No pun intended there. Is it true, do you feel that your progress is limited in any way by the rate at which protein structures, so just structure for the moment, not the dynamics, by the rate at which structures are solved, new structures are determined? The sequencing seems to be a very rapid business.
Right.
So we always know the sequence
Right.
more or less,
So
right?
that's true.
But the
because
structure determination is that.
that's solved now, though, right?
Mm-hmm.
Recently, I was just telling you guys about it before. What happened is, yes, structure was a limiting factor, and that's why there was a lot of investment with the funding agencies to get pretty structure, like making femtosecond beams and so forth. But turned out that, but a lot of people like us also try to predict structure from this kind of force field immunization, and then meanwhile, number of structures in a database got so big, and with the enhancement in speed of computer, this artificial AI, like machine learning methods, neural nets, that from the sequence, what you can do is you can look at that positions that change, co-evolve together, like making a PATS model. You know, PATS, like, right? You can use a PATS model instead of having 2D, like a spin, make it 20D because you have 20 amino acids. So you can make a PATS model. And from that PATS model of alignment of those sequences, you can find co-evolve, like positions which are in contact in space. And then you can solve structure and AI, like the people build this, and then AI take it and take it a notch. Now we have those servers can predict structure pretty rapidly. So now nothing can limit us. Instead, we have a sequence, we can have a structure, at the speed of computer, we can do the dynamics.
So if I understand you right, you're saying the rate at which experimentalists can determine new protein structures or modifications of protein is not limiting you.
Right, because we can computationally predict now.
Okay.
And they're very accurate. And actually, the whole field of structural biology is hurting because of that progress.
Say, what was that again? The whole field?
Of structural biology,
Yes.
that determine structure is being hurt at the moment because there's the software built by Google is doing it.
You know, also electron crystallography, no? Cryo-electron crystallography.
Yeah, it is getting very fast too, but still cryo-EM takes time. But if you have a sequence, give that alpha fold or omega fold, it will give you the 3D structure like in 10 minutes.
But Professor Yachman is not referring to cryo-EM, he's talking about electron diffraction.
Right, cryo-EM is the electron diffraction. You can do electron microscopy. Actually, it took over electron X-ray crystallography nowadays. If you have a super big protein, that's the limit. If you have small protein resolution, you can't do the high resolution.
You don't need to crystallize it more than a nanocrystal.
Yeah, you can make a tiny nanocrystal and get cryo-EM is also very good. A lot of big proteins will still doing, but turn out that the artificial intelligence is keeping up those experimental methods. There's the big acceleration from that end, which is very exciting too.
But today was announced that the ASU got $90 million from NSF to complete the free electron laser. So you will have a tabletop free electron.
Correct, yes, actually, yes. I'm in that center and the people in that center is building that. That's right, that's very exciting, yeah. But unfortunately, I think the idea with cryo, that femtosecond X-ray crystallography is not get the structure, actually get the dynamics at the same time. Hopefully with different crystal, with different space, that if they go to single molecule, they can get dynamics. And that's why they get that 90 million, that's the promise actually, not just the structure. All right, thank you so much, those are awesome questions.